My name is Jair Eckmeyer. I'm the Network Relation Manager at Stibion Biobank, and my profession is in the business development and optimization in healthcare organizations. Before we begin, I would like to walk you through today's program and share what you can expect from this session. First of all, I would like to introduce ourselves. My colleague, Johan, Chairman and Founder of Urimon Study and Stibion Biobank. And he's going to tell more about himself and his career and after my part. Myself, I'm a Network and Relation Manager, like I said before. I would like to walk through what you can expect today in this webinar. First of all, we start with chapter 1. That's why we started the Stibion Biobank. We're going to tell more about our vision, how we built a real-world cohort biobank for early disease detection. The second part is how Stibion Biobank works. How do we recruit our participants, and how do we manage our data? The third part is about the tools and technology. Which tools do we use and what technology do we use to ensure the right sample for the right research through our integrated systems. Of course, we're going to tell you about our collaborators, requests, and some use cases, who we work with, for example, across academia, healthcare organizations, and industry. And the last chapter is about the recap and the outlook, key takeaways, and what's next for Stibion.
I am Johan. I have a background as a molecular biologist, science, very academic career. Started with a PhD in Utrecht, did a classical postdoc in the US, came back to the Netherlands as a PI. And at some point during my research also became more interested in the more applied aspects of our profession. And that particularly started with a PhD student in the lab who started studying microRNAs and how microRNAs can affect tissue organization and tissue integrity, basically. So we found some effects of microRNAs on the integrity of tissues that we stimulated with HGF to either grow or become invasive. And all these processes turned out to be regulated by small RNAs. This was the early days. We now know that all organs contain small RNAs, and that small RNAs are keeping basically processes in check and guiding processes, and also are activated or transcribed when processes go wrong. So based on all of this research, at some point I decided that it would be interesting to see if we can use microRNAs for spying actually on the health of tissues. That's my background. I started a company together with partners for that called You to Yourself. So this is also maybe, I guess, my disclosures. But the company started with this idea, but we very soon found out it was not so easy to study early stages of the disease because we had a lack of samples, and that's why we also started this biobank, basically around the same time as the company. But the biobank and the company are actually separate entities, and here I'm talking now on behalf of the biobank. We were on a mission with the company to help transform reactive healthcare as we have it today to more proactive healthcare. And the problem is that currently diseases are detected in a late stage. When you get physical complaints, you feel uneasy, you go to a doctor, but often it's too late. We're already in the stage 3-4 disease. Things become difficult. There's smaller chances of cure. Treatment becomes heavy for a patient and also expensive for society. And the solution we think is there because we do have early signs of disease, even if we don't feel them, because we have the opportunity these days to collect biomarkers from blood, also from urine. And if we understand these biomarkers better, we can really prevent detecting disease at a late stage. But we also have obstacles. And the obstacle that we ran into and that we're trying to solve here is the lack of samples to study these signals. So samples generally are taken at academic hospitals. That's not necessarily where people present with an early stage of disease. So many biobanks contain late stage disease samples. Healthy controls is in general an issue. And And we like to use urine, and urine is really not available in large quantities in many biobanks. So that even though people pee a lot of urine, of course, every day, the storage of it is more difficult and more costly. And obviously, hospitals typically see patients only once, unless things are really wrong, and they see them many times. But to really get longitudinal sample series where you can study the dynamics of disease and really understand what it means to detect disease early was difficult. So that's why we started the Stibion biobank, or actually we started the urimon study. So we started a cohort of people and we follow them over time, and Stibion is just the organization that governs these samples and does this process. So the urimon study is a study in which we sample healthy people over time. The aim is to have 11,000 donors participate for at least 2 years. They will donate urine every 4 months, and they will donate blood every year, and they will fill out 4 monthly health questionnaires, in which they tell us whether they're healthy or sick. And when they tell us they've had a diagnosis, we contact a general practitioner for all the clinical information, but we are also linked now to the National Registry for Cancer. And we're trying to build that also with hospital registries. That's a little bit more difficult. And what we've experienced is that about 20% of these people have a chronic disease. We didn't really think about it in the beginning. And about 8% actually of the people that participate for 2 years in this age group develop some form of cancer, cardiovascular disease, neurodegenerative disease. And as I said before, the biobank makes all these samples and data available under a broad informed consent. So this was all approved by the ethical committee of the University Medical Center Utrecht. So that includes the study and the biobank. Just a little bit about blood versus urine. I assume that most of you are quite familiar with these body fluids, but there are some distinctions that I would like to emphasize, and also to explain a bit why we are so interested in urine. So obviously the filtration happens in the kidneys, but not everything gets filtered out, right? Things do go into urine, and specifically you can find smaller molecules, but also rod-like molecules, metabolic products in urine. The microRNAs that we are interested in actually end up in urine because they have this rod-like shape. But you can also do a fragmentomics when it comes to cell-circulating tumor DNA, but also fragments of proteins, of course, that are of interest. So I guess this is basically what I wanted to emphasize or show a little bit, that blood and urine are different substances. Blood is a little bit more difficult to get, but definitely a lot more difficult to handle because things get degraded easily. Urine is, in that sense, easier to collect and process, and therefore becomes also more widely applicable. And so I was given the opportunity to ask you a few questions as well, and this is a good time to do that. There's 4 of them, very quick, but then at least I know who I'm talking to and whether how this actually impacts you. So the first question is whether you're from industry or academia, just to have a bit of an impression. I guess I'm from both. More industry than academia, a little bit more. And then there's 2 more questions. I'm not sure if you already see them. They're there. So what biomarkers are you using? Protein mostly, nucleic acids, metabolites, other. Okay, so more proteins than nucleic acids. And whether it's easy to acquire urine samples for your R&D products. I guess that doesn't apply to many of you then, but it's about 50-50. So yes, no. Okay, well, that gives me a very good impression. Just going to tell you now how we work at Stibion, how our cohort is developing, where we are at. We started this in 2019, actually. I think we were into this a couple of months, and then our first lockdown for Corona started. We had a couple of those, so we had a slow start. But at some point, we started understanding how to communicate with people and how to get them interested and involved, and we started growing rapidly. And up until 2023, we grew pretty hard, a little bit too hard for our finances. So we halted the growth a little bit. You can see that with that bump over there. But we have a very loyal, it turned out that we have a very loyal group of participants. We had anticipated that people would quit participating after 2 years. That was also what they signed up for. And then by the end of the 2 years, we asked them, would you participate for another period? And it turns out that 80 to 90% is actually very happy to participate for another 2-year period. And we have been doing that now for a while. And we now have some people that are already participating in their 4th period. So in order to get these 11,000 2-year participations, we don't need actually 11,000 people, because we can count some people for 3 or even 4 participations. So that's all organized by Bram, whose picture you see over there. And we're in the Netherlands. We cover a specific part very strongly because that's where we started and where we have good agreements with phlebotomy stations. But since we did the urine now over the mail, we're expanding actually now to the rest of the country. And we do that, we can participate, we can work with all these people because we have a good digital system that works with an app. It's easy for the people. They can collect urine at home. It gets mailed to them. They have a 5-minute bike ride to the phlebotomy station, get blood drawn, and we work with certified laboratories to process and store these body fluids. And we also have a data center where we couple general practitioner records, cancer registry records, and pathology records. And all of that then is basically the core of what Stibion has to offer. So this is Stibion. And obviously we store everything according to international standards, and it's all temperature logged. So then what's in store? I guess that's the next question. What do we offer at Stibion? Well, there's our platform. We built it ourselves. The picture of this guy on the left, Jeffrey. We built a communication app to communicate with a very active participant base, actually. So we can ask new questions. We can ask them to participate in small pilots, things like that. They tell us about their health with the questionnaire. And we've built a system that we call BIMSys, Biobank Information Management System, to make sure that in all these different centers where our samples are collected, that everything goes exactly in the same way, and we can trace back everything that happened. So it at least helped us to cut a lot of our paper waste. And it also helps us or enables us with only a very small team, basically Bram, 1 technician and an assistant, to manage all of these people and all of these samples. And this platform is actually, we designed it for what we want to do, but we feel that this is useful and transportable to other initiatives as well. So we'd be very happy to share resources here. Little highlight is BIMSys, because we're just developed it and we're really happy with it. And it's also the feedback that we get from other users. It's really about being able to trace samples across multiple different sites without any mismatches in compatibility, losing samples, not knowing exactly what happened to a sample. It's a new system that we're very happy about. And then obviously the most important thing, I guess, since we're talking about biobanking, is samples. Our biobank is a cross-section through the Netherlands. We have the obvious risk groups. We have the people that are drinking heavily, the people that are smoking heavily. We have people that have severe obesity, and we have a lot of people with chronic diseases, as you can see in the right bottom there, that also develop other types of diseases because of that, of course. This is current data. We put these numbers together yesterday. So that is there and growing, and obviously the most interesting for ourselves, but also I think that is unique in our biobank, is those early disease stages where you can really either capture the onset of the dynamics of early disease. You can see the numbers yourself. We have over 2,000 of new cases. It's actually incredible how many diseases occur. We focus on cancer and cardiovascular diseases, and cancer is, of course, half of this is skin cancer. So that's I guess, less interesting when you think about liquid biopsy diagnostics. But the solid cancers and the blood-based cancers are also growing. The numbers, you can see there on the right. So this is just another way of representing the long series of samples that we have. So these are sample series that are at least 4 samples long. That's also growing on us so that we can really see those early stages of disease. Well, we get requests from academics, startup companies, smaller, larger companies that are developing instruments. We aim to participate in public-private partnerships and these big European partnerships, of course. And recently, the government also found us because they're interested in doing biomonitoring to track toxicities in the population. And just to show you 2 use cases that we did with the biobank, this is our own use case, the microRNAs. And we did a proof of concept study there in about 400 samples where we could really see that these people that were going to remain healthy, wasn't known at the time of sampling, can really be separated from the people that were going to be diagnosed with a disease. This is done here for 8 different diseases, or 9 actually. And as a result of this particular experiment that we would not be able to do without the biobank, we got some good investments in the company. So the company is benefiting from this as well. But that's just a use case in this case. The other use case is the Netherlands Institute of Health that are doing a toxicology study for food safety, and they're relating it to kidney function. And that looked so good that we're actually talking with them about developing a national program for this. Well, that's actually what I had to say. I don't think we need a recap right now. You've seen it. There's a few minutes for questions. That's important. So let's go to the questions. Thank you very much for your attention.
Okay, fantastic. Thank you very much, Johan. What an amazingly valuable initiative. Just looking at the Q&As. Okay. I don't see any questions yet about the
It's all about the screen.
content. Let's have a look. Okay, yes, I do. Okay, there's a question from Ombretta Colasanti. Do you also have other biosamples other than urine and blood?
No, we do not. We started urine and blood because we thought those were the 2 main accessible samples in a general real-world setting. We have been talking about including things like saliva and stuff, but that's such a logistic change that I don't think that that's going to be feasible anytime soon. So it will stay urine and blood for now. Yeah.
Right. And we have a question from Riyadh Tebaka. You said urine's collected from fasting patients?
I did not
or at any
mention that.
time?
No, I didn't mention that. No, we don't do fasting. We do first morning, first catch, and first morning midstream urine. That's what we do. So we ask people to let a little bit of urine go in the toilet and then collect the urine in the collection cup. First thing in the morning. Yeah.
Jennifer Marks asks, since you are collecting urine from multiple locations, how can you be certain that the samples are handled in the same way before processing?
Yeah, that's a good question. So we specifically for that we built our biobank information system. So everybody who is processing sampling for us, doesn't matter in which organization, is using our software. So it's web-based. So everybody can use it with an internet connection. We log everything. So every time a sample gets scanned, so they get instruction on the screen. I think that works pretty well. Obviously, we do some quality checks and we never see big problems. And good question for the second one. We use a preservative, and that is already present in the collection cup. So at home, the people pee in a cup. there's a preservative that contains EDTA, azide, and a little bit of glycerol, and that basically preserves the nucleic acid biomarkers that we are interested in. The proteins we haven't studied sufficiently to be able to say that as well. And metabolomics usually works well. Yeah.
Okay. How many pancreatic cancer samples? Asks Malcolm Mackenzie.
Pancreatic, I believe 2. It's not a very common disease. Nobody was diagnosed with it. So no. And non-cancer controls, thousands. So that's an easy answer.
Okay.
Yeah.
Question from Naveen Kumar. What were the key challenges from the regulatory side to establish this biobank?
The key challenges from the regulatory side, well, in the Netherlands, the ethical approval is about a study. So you have to do a study with a purpose, with the right power, all that kind of thing. It's a risk-benefit thing that the ethical committee has to decide on. So it wasn't very difficult. We wrote the study protocol, we had it discussed and approved, and then the biobank was a secondary thing. I think the rules in Europe are now changing. There's going to be biobank laws, but there isn't one at the present time. So it all depends on the study currently.
Right, right. A question from Malcolm Mackenzie. How does one access sample sets? Via collaboration or by purchase?
It can be either way. Purchase is the easiest. You have to be within our goal, otherwise we fall outside of the informed consent. So early disease detection, health monitoring, it's a broad scope. So usually we're on the same emissions here, so that's okay. So yeah, you can just contact us and we'll set it up. Can be either just direct purchase or a collaboration.
Okay, okay. A question from Warren Fransman. Do you have a sample access committee approving or rejecting requests?
Yes. Well, we have a process. If it's a few samples, then we don't go through a complex thing. If it's a large project requiring a lot of samples, then we go back to the ethical committee of the university, and they actually have to approve.
Great questions. Thank you, everyone, for those.
Thank you very much.
And thank you, Johan. Very interesting presentation. Thank you very much.
Thank you, everybody.
Thank you, everybody. Bye-bye.